is Tuesday, August 11th, and you are watching the Biotech Voyager. It's the show that covers what's next in biotech. We're covering the early stage signals before anyone else is. The really cool science is going to be a big deal in a couple years. We've got a very exciting show planned for today. We're going to be starting off with a guest. Normally we talk about the news for about half an hour first and then bring on guests, but we've got a very special guest who is only available at 11 today here in Eastern Standard Time. Very interesting guest who's focusing on a PhD program.
Doctorate program.
A doctorate program, yes, that basically delivers a full startup at the end of your doctorate. So we're really excited to kind of get into that in more detail in just a minute here. We've also got some great signals to cover today, AI designed molecules, some really cool moves inside of the siRNA space, some big moves inside of a company that we've talked about,
Dispatch Bio
in the past, one that Jeff has
opinions on.
We'll get into
Shocker.
We'll get into all that and more. But before that, let me tell you about Tetri Bio. It's all about who you know, but now you can catch prospects the moment they're ready. Deep company research, live signals from your socials, verified contacts, and a whole lot more. Built for biotech and no one else. That's Tetri.bio. That's something that we have developed, Jeff. That's a company that we run. So
it is. Based on the word Tetris for being the perfect fit.
That's right. Excellent. All right. So, Jeff, let's get into this interview, shall we?
Bring him in.
Excellent. All right. So we're very excited to have Dr. Thane Campbell with us. He's the Dean of Education at Deep Science Ventures. And so with no further ado, let's bring him on the show. Thane, welcome to the show.
Guys, thanks so much for the drum roll.
Normally Ben's time's a little bit better than that. A little bit early this time, but let's slow it down next time.
Excellent. All right, so yeah, Thane, we'd love to get a little bit of your background first. What kind of brought you into Deep Science Ventures? What's your background? What brought you to where we're at today?
Yeah, thanks. And so my journey into this whole sort of PhD reformation space kicks off with my own PhD. So I was at the University of Edinburgh. I was studying immunology and AI. This was before COVID and before GPT. So before these things became sort of global phenomena. But still fascinating work for me. So 3 universities and 2 large companies all working on a core challenge of how we interpret the signals from a number of lung diseases and diagnose them, tell them apart so that we can actually treat them. And so it's a thrilling space for me to get around medical imaging, but also really work with signal processing people and chemists, a kind of complex space that is not hugely typical of the PhD. And during that time, I encountered Deep Science Ventures. And this was like an extraordinary moment for me. I regularly talk about it as my Plato's cave moment because it was a room full of scientists who were all fixated not on like, how do we get more positive data and publish? Good though that be for the majority of the scientific enterprise, but it was really stimulating to be in a room where people were trying to answer the hardest and most important questions in their industry and in their field. And as they landed on things, getting companies built around them, getting industrial partnerships, funding, and just watching the thing grow and scale in real time was really, really fascinating. So I knew something then from my own PhD days had clicked deeply with this model. And then increasingly it seemed important to do something at a PhD program scale. And that's where we are today.
Right on. Fantastic. Jeff, you're the PhD in the room, so
yeah, so I don't want to jump too far ahead, but a couple of the things that I found surprising when I finished my PhD studies, because your like epiphany Plato's cave moment happened during your PhD. Mine happened like 3 years after my PhD. And I realized that my PhD, I was very sheltered during my actual graduate studies. And I'm not super sure if it's because of the institution that I got my degree from or what, but they were very, very, very either against or just like averse to commercialization. It was very like basic primary research driven, academic career path driven. And for me, and I feel like it's probably very similar with you, I did not resonate with that model. I always compare it to playing in the sandbox, right? Where it's like the people who care about my research are the ones who are being paid to care, like my advisor and my committee and things like that. Nobody else really cares. And I didn't know how to shake like the turmoil of that. And it sucked, right?
Right.
But if I would have known that, like, hey, you have the ability, the opportunity to actually take this and turn your current thing into a company, or, you know, it can become its own thing, that would have changed how I performed the experiments, how I designed my research. Like, it would have changed everything. So it seems like this is desperately needed for a lot of people in their graduate studies.
It's so funny hearing you talk about it because so much resonates. Like, it literally did change how I did the rest of my PhD. So I came back from Deep Science Ventures. I completed my PhD in a completely different mindset. First of all, it was bold enough of the people who designed my PhD to like leave 3 months to just go anywhere and work with any company. And like coming back, I realized that there was so much opportunity, so much of the world wanting to see PhDs and scientists like aim at global challenges, dissect them, and find things that could scale as solutions. That then I looked at a number of experiments that I'd been doing differently and realized that they could kind of plug together. And so there was a point towards the end of my own PhD where I had an invention, and a bit like you, I saw how frustrating it can be to try and like move that against the brick walls that you can then come up against in terms of translation. And so I don't think about this in terms of people doing a bad job or like anything at that level. I think we fundamentally have systems that were designed for getting people academic tenure. And there's been work to try and change the model or add things to the model. And in some instances, we're just pushing far too much on academic departments to try and be and do all things. And in the other instances, you have to think about tech transfer. Like, you've got 40, maybe 100 people trying to service the invention portfolios of thousands of, you know, so like the systems that we found ourselves in to bring change into the world were not designed to spool out elite deep tech founders. So we had to think seriously about how to do that differently. And it's just... Do
you think that there is something unique about the life sciences sector in particular? And it's why I'm actually very interested to know your opinion on this particular question. Is there something in particular about the life science space that makes this problem more potent? Because typically you need to have, like for me, like in a restaurant business, for example, or even in like, I don't know, pick any other like science, or not even science, but like pick any other business sector. The people who run the companies don't have to have a PhD in some deep science to do it properly, right? But when it comes to like biotech, for example, typically like the technology itself is coming out of, or the people, the technology has come from PhDs, or the people who are running the companies are directly themselves PhDs. Whereas I don't think it's that common in other spaces.
Yeah, sure. So it's an interesting one to focus on life sciences and bio. And so I think after defense, pharma is like probably the most funded industry in the world, and that's got trickle down effects on like how many PhDs we can train. There is some interesting work that the National Academies of Sciences did, where they anticipated, it's in the Bridges to Independence report, that the field of biomedical sciences would be lost to the US because of how the PhD was creating like bad news for people behind them. So like the more people who go through the PhD and don't get the job that they'd spent 5 to 10 years working extremely hard to get, telling the story behind them. And before I joined my PhD, this was a thing. Like people were telling me, like, don't do it, run away,
Right.
do anything else, go
Right.
and live a happy life. You know, like don't sign up for the
go to med school was my advice.
Right.
Choose
right.
joy. Yeah.
So I think there's something in the life sciences where the sheer scale of the number of people going through different biological, different biotech courses, and then experiencing this kind of amplifies the news around it. But the main thing is that there are many industries where you need a PhD to be able to invent at the cutting edge. So clean energy, nuclear fusion, semiconductors, AI now. There are loads of these spaces. There's loads of ag tech innovation that requires a lot of heavy research. And what you fundamentally need is a process to systematically invent, to find like what outcomes do we need in the world and how can we map those onto a set of research projects that would get us there.
Nice. So yes, basically take us through, right? Like the main thing we want to focus on with this interview, right, is this venture science doctorate. So
Sure.
How does this work? How does someone sign up? What is the process by which someone who's considering a standard PhD, right? They're thinking they might go to grad school. It's a little bit of a difficult proposition because the job prospects in a lot of these different fields isn't as clear as it might have been 20 years ago. And so how does this venture science doctorate work?
So the key thing first is Deep Science Ventures actually delivers this program. Mm-hmm. We are a venture creator. So we're not an incubator, we're not an accelerator. We're a place where people start working on hard problems and leave with the company. We've built 60 deep tech companies. They're worth $1.3 billion, and they're backed by Breakthrough Energy Ventures, Sam Altman, Sequoia Capital, and others. And so everything that we've done to build that deep tech portfolio in ag tech, in climate, in therapeutics, and in computation, the protocols that we have for finding people for invention, for investment decisions, this is what's been folded into the venture science doctorate. This is really where it's come from. And so that PhD program today is a 3-year program. It is one that is completely focused on invention. And it's one where you start with a global challenge and you build a science company to solve it. So we've had to think about recruitment fundamentally differently because one of the things that we realized while we were still designing this was if we get this right, and if people can walk into universities and leave with a deep tech startup, then another thing that we absolutely have to get right is the diversity of people who are doing this and who can access these spaces. And so recruitment became, like, logically and philosophically the first most important question for us. And so we recruit around magnetism, expertise, and determination. And each of those things breaks into 3 subcategories, and each of the subcategories have indicators. And so magnetism is concise storytelling, persuasion, and empathy. And for each of those things, we're measuring things. And so this is our definition. Those 9 categories are our definition of what we think the ideal science entrepreneur is, and how they function and how they think and some of their attitudes. And those are the people that we're looking for when we start a round of recruitment. We've just closed one. And then in the program itself, it's 3 years. In the first year, you go through quite a rigorous process for designing your venture. And this, I've touched on it already, but you have to aim at a societal outcome. And there's ways to systematically work backwards from that. The second year is really about now moving that into prototyping. And so how do we make the real invention of the thing that you posited? And the third year is closing out that research, and bringing on your co-founder and securing investment. So that's kind of what the track looks and feels like across the 3 years.
So that's interesting though to me, because what do you do you get this doctorate degree on top of like a standard PhD? Or because like for me, my PhD was 6.5 years and it was spent 5.5 of them were in lab learning how to dissect mouse brains. So like where does that come in?
Yeah, this is the thing. It's a fundamental reimagining. And so what we aim to do here is not to compress 6 years into like 2 years of prototyping and R&D, but to actually enable people to move with a lot more fluidity into the field that they need to gain mastery there, to create the data set and the proof of concept and the prototype that persuades investment. And then to have the vehicle that itself, the venture, continues doing its research. And so it's not a question of like gaining all of the mastery of people who are going for tenure. But actually it's a different question entirely of how do you invent? And so for example,
yeah, exactly.
Vlad, who came into our program, his master's was in architecture, and he's building a new nuclear fusion reactor architecture. And so he had no prior experience in fusion. So he was telling us these stories about how he's designing it from first principles and using all of our scoping protocol, which is how we invent. And we were all thrilled for him because it sounded great, but you're always asking yourself, my background is not in fusion either. And so is this really going to pan out? Is he really hitting on something? He recently secured 200,000 euros from the Federal Agency for Breakthrough Innovation in Germany, Sprint. And so this grant, their validation grant, is something that they usually only give to professors working on the most important sort of frontier problems in the world. So it turns out that what he was doing stacks up. And he's now leading a consortium funding PhDs in the universities of Alabama and the University of Rochester, and working with one of the co-founders of Commonwealth Fusion, which has raised nearly $3 billion. And so like, that's the question. How do you get people in a position where they have total autonomy from day one to achieve the maximum conceivable impact on an industry? And Vlad was looking at clean energy, and he realized that, renewables are fantastic, but they're not consistent, reliable enough to displace fossil fuels. So he started asking questions around clean energy that never sleeps, that has this ultimate reliability. And it pushed him.
Amen. So many of these, this is a tangent, like so many renewables, it's like, come on guys, like wind and sun, they're the main attributes of them is that they are unreliable sources. They ebb and flow. Like if we want to reduce our dependence on fossil fuels, nuclear is the only way. So anyway, sorry, continue.
Yeah, no, there's going to be a portfolio of approaches for sure. And I'd love to see renewables continue. I'm from the Bahamas. Originally. So I'd love to see solar and other renewables pick up and be connected to things that change their paradigms. But for sure, we're going to have to ask these hard questions around where do we find scalable solutions? And in fact, like what he was doing in particular, fusion today, there's real questions around even if that will scale when we look at tokamaks, when we look at stellators. Because the designs of these reactors are so complicated that it's unlikely that more than a few countries will ever make them. They rely on tritium, of which the world has 25 kilograms. And so Vlad's design, when he was inventing, he specifically went for simplicity of that design. He specifically went towards deuterium, which we have a quadrillion times more of.
Yeah.
And so like this is what we're trying to get people to do, is to like fundamentally rethink these hard questions that the world is, like clean energy.
So stop me if this is like not relevant, but the idea that you need to have like deep I'm a biologist, right? So the idea you need to have this like deep laboratory experience and the ability to like perform the experiments yourself in order to run a biotech company. It's like so not true. Like when I learned about what a CRO was, I had like a heart attack. I was like, wait a minute, I can pay someone to just do my experiments for me? Like, wait. That changes everything. Like, because it's like, if I couldn't do the experiment, I just like didn't ask the question, right?
Yeah,
so the idea that like,
the core
yeah,
skills
you
are
go.
going to increasingly be around like problem definition,
Mhm.
product specification, and fundraising. These things
Yeah.
really change the narrative. And so you can fund someone to do the research for you. Then it becomes more important how you choose that research,
Yeah.
and how you design it so that so if we actually want things to become products in the world, there's a whole set of questions that we have to ask and answer that we wouldn't if we were just doing basic research. And so if
Mhm.
you want your research to become a technology that can then scale up, and reach the later technology readiness levels,
Right.
then you'd have to design your first experiments differently.
Yeah, exactly.
And so it's increasingly around like, which problems should we answer and how do we answer them in a way that can lead us to products. We do have people in the program working more on the closer towards the kind of asking fundamental biological questions. And so Daniela is in the program, building a solution for female health. What I love about what she's doing is like, so her solution will extend the health span of women by 5 years. She's doing this by rejuvenating the ovary, essentially pushing back the age of menopause. And crucially, like, when she started scoping this problem, she realized that she landed on a solution that's deep to both female health span extension and fertility. And so these are two huge questions when you think about the fact that like workforces in Europe are shrinking because of fertility problems, and the age at which people are having children later in life. These are both huge unmet needs in the world that we need more research and technologies around. And to find something at the nexus of both of them is really phenomenal. And so Daniela is now collaborating with some quite prestigious labs at Harvard to get her research done. And this is and they will support the funding of that research and these questions that you raise around like how we get the work done itself. And so again, if you land on something that's so important, it's compelling to some of the most entrepreneurial researchers in the world, then what you need to do next is not, do all the experiments yourself. You need to think about who you can work with to generate the best opportunity for that research and for us, a venture.
Yeah, that's really interesting, right? Because so often in PhD programs, to Jeff's point, right, it's very much you are trained to think and operate like a single operator, and that's kind of it. Versus, if I understand correctly, from the get-go, you guys are very much in the position where you're getting these people to start thinking a bit more big picture and to start orchestrating the labor of other groups to help achieve that end goal, as opposed to trying to do it all yourself. It's very much putting them almost like one layer up of coming in and starting to move all these different pieces together, as opposed to being the individual contributor.
Yeah, I think this is one of the big fundamental differences. The PhD is sort of, if we use broad strokes terms, traditionally it's trained someone who's a very individualistic researcher. They're going to produce new knowledge and ideally train a lot of people after them. And we thought we kind of need to broaden the definition of scientist a little bit, because we are saturating those jobs. What we need is a kind of demand side scientist who can produce new applications for that knowledge and create the jobs that these people will be absorbed into. So when we're thinking about like the future of the biotech workforce, like we're not going to employ all of the PhDs who are being trained today in even that industry. So there's a huge opportunity to kind of change the nature of what we're asking of scientists so that they land on scalable companies that create opportunities for more fundamental research, more science jobs behind them.
So how do you find these people? How do you
Yeah.
what do you guys do?
Yeah, so at the moment we simply like put the word out on our socials and like get maybe an article or two going, and then we've been surprised at the number of people who will put their hands up for this kind of thing. So we've had over 3,000 applicants over the last couple of years, and
Nice.
and have taken we have 14 people in the program, and so like from a given cohort, we're taking like the top 0.5, 0.3% of applicants, and they apply from like over 100 countries.
Is it in person? Like, do you got to go to the UK?
You don't, no. So Deep Science Ventures itself is like decentralized, and we have companies in over 24 countries now, I think. We have people working in Kenya and in Europe and in the States. Like it's a hybrid program in that you can do a lot of the learning in a virtual environment, and then you're moving into labs in a network of 40 universities that we're working with, which includes like Cornell Tech, Imperial College London, the Helmholtz Association in Germany, which is Europe's largest research asset, and several more. And so the whole thing is designed to give you flexibility to act where you need to build the company that matters the most.
Love it. Okay, so yeah, like 0.3% of applicants get accepted. The top 0.3%, of course. And so like what are those evaluation criteria looking at? Is it kind of that evaluation of those 9 different things that you mentioned previously?
Yeah. So if people come with some background in startup or VC, that tends to help them to think around invention or a kind of portfolio approach to things. We really need people to be idea agnostic. So we have a process that you would use to like step through systematically building out like a tournament of ideas and then comparing and ranking those. And so the idea is that, even if you're working on
tournament of ideas, that's
like a bracket.
That's poetic.
Yeah, no, I mean, if you think about it, after this program, and even in the last year, you're pitching to a bunch of VCs, and they see many, many different people in their category all the time. And so you have to anticipate how you're going to stand way out from all of that competition. And a good way to do that is to start to like run your own competition of approaches towards what you would want to build, and start to think about like what differentiation factors look like in your industry.
Super cool. All right, I know we're almost at time, so we do want to be really respectful of your time. But I guess like one other question at least would be, well, first of all, do you think that this is something that just every venture studio should start doing? Is this kind of like a new arm that should open up? Are they collaborating directly with a specific university, and or are they awarding the doctorate themselves? What's kind of the process there? Is this something that could be applied to other venture studios?
What's really interesting is right now in the UK, the government has just announced a call for venture doctorates. So these kinds of methodologies are going to be increasingly brought on campus. It's a great opportunity for venture studios to work with universities to create these pipelines of deep tech companies. And it's something that I think we're going to see more governments think about in Europe, in the US, and in terms of, if we're spending billions every year on PhD research, maybe some fraction of that. So today, the fraction of people that invent and spin out in Europe from STEM PhD is 0.036.
Jeepers.
And so like maybe it should be 0.1, you know, like maybe it shouldn't be. There's a huge opportunity there, and it's an opportunity for academia as well, because if your PhDs spin out, draw VC investment, scale, and they have good relationships with the university, this is very good, as we're seeing from some of the universities on the coasts.
Fantastic.
I wonder why that's so like entrenched in academia though, because it isn't a controversial thing to say we should try and commercialize this. Like what else is the point? But I feel like academia has been so, I don't know, it probably goes back like hundreds of years. It's like it's a very closed system.
I think there's actually, it's kind of like the very famous like Charlie Munger clip. He was like the guy who who co-founded like Berkshire Hathaway with Warren Buffett, and he kind of has this famous line along the lines of, you know, the thing with incentives is you get what you reward. And so, right, like the entire academic industry is very much kind of focused around like what gets the next grant. There's not the incentive structure that allows for spinouts into industry. And so potentially, right, the incentive here would be for venture studios to come in and partner with those universities, thereby providing the reward for that kind of behavior. Yeah. Awesome. Perfect. Well, Thane, really appreciate your time. Again, we want to be super respectful. We know you gave us a couple extra minutes. Thank you so much. Really appreciate your time. We're really excited to see what Deep Science Ventures does with this program. And yeah, maybe at some point we could have a special episode of the Biotech Voyager where we bring some of your different cohort members on. We'd love to do that with you guys.
Sounds fun. Excellent. Thanks so much for having me, guys.
Oh, thank you so much.
Thanks, Thane.
All right. Right on. Jeff, don't you wish you'd known about that 8 years ago?
Like the thing too is that there's not everybody who gets their PhD would want to do this. There's
right.
people who are like, I love doing lab work. I want to be a lab tech or whatever, right? But there's a good proportion who are just like, this is not what I thought it was going to be, right? I would have been one of those people that was like, just like drooling over this program. I would have been like, absolutely, give me some. Right. So yeah, I wish that I would have known, but I'm very glad that one, it worked out anyways. Ben, I wouldn't be here if it wasn't for my PhD. So it led me to you. What do you know? But at the same time, like there's plenty of people who are going to benefit greatly from that kind of program. So looking forward to see what they do.
Oh man, this should be something we try to let's see if we could help out with this, Jeff, because I would love to just start like bullying some of the different people that we know inside of different venture studios and be like, go find a university and, you know, implement the blueprint that Deep Science Ventures is doing. Get ahead of the curve. Mm-hmm. Make your own future portfolio as opposed to kind of waiting for people to self-select in. Like there's got to be such an interesting like return on investment there, eh? Of being able to kind of get in on the ground floor of creating your future portfolios. It's got to be there's got to be a significantly higher rate of return on something like that.
Out of my pay grade.
Excellent. Okay, very good. So yes, ladies and gentlemen, we just had a chance to interview Dr. Thane Campbell over at Deep Science Ventures. He's the Dean of Education there. If you're just joining now, feel free to go back and view the interview we just had with him. Really interesting application of basically developing a venture science doctorate, where instead of kind of going down the single operator path that is a standard academic PhD, the goal is, by the end of your PhD, you're going to have a company that you can start and implement, and they've had some really interesting success so far. So feel free to go back and look at that. Okay, so Jeff, the early stage leaderboard review. Can we just go back to the leaderboard itself for a second? Mm-hmm. What's right at the top here? Expedition Therapeutics. Right on.
Yeah, so it's been kind of a slow like beginning of the week here. To be honest, one of the most interesting things that I've seen was this LifeMind Therapeutics. I think that Biotech TV interviewed them. They had a massive like late stage raise. Nice. They're doing stuff with like organ transplantation and things like that, like helping organ transplant recipients. Like I don't know. Oh wait a minute. This is totally not. Is it that? Hang on a second. I'm almost positive this is what it is. Yeah. Transplant rejection. What are they doing with? Why did I see bacteria? Wait, what?
Fungal genomes. I saw this one too. This is super cool,
LifeMine.
Jeff.
Okay, mine's fungal genomes using its proprietary Avatar RX platform, integrating genomics, AI, and synthetic biology to discover genetically encoded small molecules inaccessible to conventional drug discovery. Its lead asset, Life 001, is a precision-engineered immunophilin-independent calcineurin, and I should know what that word is. Calcineurin inhibitor targeting immune-mediated disorders and organ transplant rejection. That's crazy. I'm almost positive that the first antibiotic was discovered in fungus, right? Like some kind of mold.
Yeah, penicillin.
So it's kind of like I'm thinking of penicillin. Yeah.
Penicillin discovered in Canada.
Really?
Yeah.
Yeah, because it's so dirty up there.
Whoa!
All that mold growing. But yeah, right. I'm pumped to get into Dispatch Bio. I have thoughts and questions.
Excellent. Well, yeah, I do want to come back to this fungal company a little bit later. But yeah, in the meantime,
did you add them?
I don't know if I did or not.
Okay, we can come back then.
But regardless, yeah, let's jump into Dispatch, Jeff. Hit me with it. What's the what's the big deal?
So the signal was they dosed their first patient in GI cancer, right?
Well, I mean, first of all, congratulations.
Yeah, and it looks like they got fast track designation in the same press release. But yeah, they came out of stealth, big blow up, like the Sean Parker Institute did a big fundraise into Dispatch, and I think that Carl June's involved in this. It's a UPenn like spin out, and what they're doing, we talked about this before, but what they do is they have this flare protein approach to CAR T cells, right? Yeah. So two of the biggest problems with CAR T cells is the immunosuppressive microenvironment with solid tumors and the lack of a consistently expressed, like widely expressed, highly expressed target antigen in solid tumors, right? In heme, you can do CD19. CD19 is a massive target in hematological malignancies because most B cells will express CD19 at a very high level. You can have the CAR T cells deplete the B cells targeting CD19, and you can just like kind of ablate the entire B cell population, including the cancerous B cells. In solid tumors, it's a different story. You need to have a consistently expressed antigen on the surface of those solid tumor cells, and it's hard to get that. So Dispatch Bio has a way around both of those problems, the lack of an antigen and immunosuppression. And it's this flare protein thing. And it's essentially a combination of it's kind of like an oncolytic virus plus a CAR T cell.
Yeah.
Well, yeah. But it's the concept of an oncolytic virus plus CAR T cells. So what they do is they have this approach where they'll infuse a adenovirus that expresses flare. It's a flare protein. It's a synthetic protein. And I think that the virus also expresses like cytokines. And what they'll do is they'll selectively infect the cancer cells with various mechanisms, cell selectivity, promoter selectivity. And they'll have the tumor cells themselves expressing the cytokines and this flare protein, right? And then they'll simultaneously send in CAR T cells that recognize flare. So they'll go and they'll kill the flare positive cells, which should be the cancer cells. So then you get the elimination of flare positive cells, and you have the expression of those cytokines overcoming both antigen, lack of antigen and immunosuppression.
Yeah.
That's the whole idea. And they're doing it in solid tumors. They have their first patient dosed in GI tract cancer. The question that I have, Ben, is why? If we are getting expression to a high enough level of flare within the tumor, we already do this with oncolytic viruses. And the oncolytic viruses, they don't make the cells express flare, they just kill the cells. So what Dispatch is doing is making the cells express flare, and then they kill the flare positive cells with a CAR T cell. And it's like why? It seems like an unnecessary added layer of complexity, you know?
So if you go to their leadership page, Jeff, if you quickly go to the Flare website, back at ASGCT, I was fortunate enough, shout out to Jay Rosenelli over Synex. He invited me to a dinner of a bunch of different people inside of the biotech space. And one of the people there was a gentleman called Lex. And
Lex he's the founder.
I believe he's like the chief science officer.
Well, it was his like research. It's this guy.
It was his research.
Guy. Yeah.
If you scroll down a little bit. That guy right there. Perfect. Lex Johnson. And so I got to sit next to him. And so I'd asked him this exact question. It's like, why? Why wouldn't you just use an oncolytic virus? And hopefully I remember correctly what he said. But what was really interesting, what he focused on was the fact that what are cancer cells really good at, Jeff?
Growing.
They're really good at growing and secreting all sorts of mechanisms to not die. And so the challenge that I believe he outlined with oncolytic viruses is the fact that cancer cells are just really, really, really good at mutating and evading all sorts of mechanisms that cause them to die or to signal to the immune system. And so the workaround here is don't try to kill them. Just try to hack the mechanisms that they are really good at doing, which is to grow and to not stop growing.
So here's what I would have said to that.
Let me finish. And so there's two different things that he focused on. There's that first component, basically how can we overcome the immune escape issue, and secondly, how can we make sure that we're using something that is going to really, really effectively infect cancer cells. Even after the first round, these adenoviruses will continue to infect the additional generations of the cancer cells, so you really get robust expression of the cancer cells. And hopefully, I'm doing his explanation justice. I might be missing something.
So if you didn't miss anything, I still don't like it. Because, and sorry that my stupid computer is not working. I'm on my phone's hotspot, so my Wi-Fi is not the best. But I can hear you. Hopefully, you can hear me.
I can.
What I would have said to that is, okay, fine. You're not directly killing the cancer cells. You're just making them express this innocuous protein. And they have no problem doing that, right? But in effect, the expression of that flare protein is killing them because it's making them a target of the CAR T cells. So any of the population of the cancer cells that don't express high levels of flare will just outcompete the ones that do, just like flare was actually killing them.
So my understanding basically is that the approach that they have taken is really, really, really good at making sure that cancer cells do express the flare.
Okay, that's fine. But if they've taken an approach that makes cancer cells really, 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 really good at expressing flare, then just make them express some kind of apoptotic protein. Like there's no way around this. Like no, it doesn't make a whole. I don't know. I can't make it make sense. There's got to be. We're obviously missing something. Like if they've said, okay, we've gotten really good at specifically targeting only the cancer cells, and we can make them express this thing at very high levels. It's like you've gotten very good at expressing something at high levels only in cancer cells, and your payload of choice was a non-lethal tag protein that you then send in CAR T cells after. Like, it just doesn't make sense to me. And maybe I've got to be missing something because like Carl June's involved and there's very smart people who have backed this and it's like out of UPenn. So I'm obviously missing something, but on the surface, it just seems like an unnecessary, like it's a cool idea, right? Like it's kind of like an alley-oop in basketball. Oh gosh, can you hear me?
I can.
Okay, my computer is like completely black now.
Yeah, but your audio is good.
It's kind of like an alley-oop in basketball, right? Like an alley-oop is cool, but don't go out of your way to do it if you can just do a layup. You know, like that's not going to win you. It's not going to do anything. You know, it's kind. Yeah, that's probably the best analogy. Like if you can just walk up there and do a layup, like what's the point of forcing yourself to do an alley-oop? More things can go wrong.
Well, let's jump into this because they've got like a science tab. Let's run through and see if they have something in here that addresses this.
Mm-hmm.
Because
if my computer will comply.
Yeah, go to the science.
Okay, so immunotherapy has revolutionized the treatment of certain blood cancers. However, increasing its impact requires expanding access to solid tumors, which represent approximately 90% of cancers worldwide. The impact of immunotherapies in solid tumors has been limited because of two things: lack of tumor-specific targets and presence of an inhibitory tumor microenvironment that impairs immune responses. Okay. Our first-class FLAIR platform is designed to overcome these two core challenges. It first dispatches a tumor-specific virus equipped with a novel universal antigen called FLAIR that tags solid tumor cells with exquisite specificity while simultaneously activating the immune system and eroding the inhibitory tumor microenvironment. The FLAIR antigen then acts as a beacon, guiding the way for the immune system to find and clear the marked cancer cells while avoiding healthy tissue. Okay, so here's something that I could think of that might be a nuanced dimension to this. You ready?
I'm ready.
FLAIR, while it will be expressed natively on the surface and can be used as a CAR T cell target, it's also a foreign peptide.
Yeah.
That will be recognized by the immune system as being foreign, like a viral particle, and it will cause the immune system to kill it independent of the CAR T cells.
Yes, I believe that is the other component that I probably forgot to mention fully. Yeah, you're basically exactly what you just said. You basically have the ability for the existing immune system to come in and kind of like serendipitously also attack cancer cells in the context of like a viral infection.
Okay, so I think that's a little bit better, but I think that like we've kind of lost the plot a little bit, because the point of making immunotherapy work is to kill cancer cells. It's not, the point of making immunotherapy work is not to make immunotherapy work, right? The ultimate end goal is to kill cancer. I think that this is like a solution to the problem of immunotherapy doesn't work in cancer or in solid tumors. So how can we make immunotherapy work in solid tumors? But the question really isn't how do we make immunotherapy work better in solid tumors? The question is how do we kill solid tumors?
Right.
You know? So yeah, I feel like, is my camera just completely frozen now?
It is, but I can still hear you.
What in the world, dude? It's so...
Have you got Claude doing something crazy?
No, nothing's open. I'm on my hotspot because I'm in my sister's house right now in Buffalo. So, but anyways, okay, you know what? I'm just gonna do this because I don't feel like being that person.
Fair enough.
Oh man, that's so annoying. Come on. Nope, still frozen. Anyways, do you see what I'm saying though? Like it does seem to me like it is a solution to the wrong problem. Like and like Carl June's a CAR T guy, right? So it's like, how do we get CAR T cells to work in solid tumors? Well, why don't they work in solid tumors? Lack of an antigen and immunosuppressive microenvironment. How do we simultaneously overcome that problem, both those problems, to make CAR T cells work in solid tumors? This is a solution to that problem. But it's like, yeah, but the solution or the problem is not to make CAR T cells work in solid tumors. The problem is solid tumors, you know? So yeah, what are your thoughts on that? I don't know, man. I still don't really see the point. I'm sorry.
My understanding is it does have quite a bit to do with the fact that it is an adenovirus that's able to actively infect the cancer cells and to bring in that endogenous immune system. But
oncolytic viruses do that. Oncolytic viruses do that.
Yes, but they don't provide. But they don't additionally provide this foreign antigen that's only going to be exquisitely, in their own words, exquisitely expressed on cancer cells.
Yes, they do. Yes, they do.
A fully unique peptide, though.
Well, it would be foreign, so it doesn't matter. It would be a foreign peptide that would overcome a tolerance. Like,
sure, but like
you're infecting them with a virus.
What would that peptide be? On an oncolytic.
The viral peptides from the virus.
But like won't most of those be endogenous inside of the cancer cell? And won't some of these?
No, they're not any more or less than FLAIR would be.
Well, no, I think the other component of FLAIR, right, is that this is a type of protein that is going to migrate to the surface and get expressed in high quality.
Yeah. So what I'm saying though is like, I think that flare will simultaneously be expressed fully and then pushed to the membrane, and it will exist as flare on the membrane, which can be targeted by the T cells, the CAR T cells, sorry, right? And then also flare will be processed and loaded onto MHC and be recognized as a foreign peptide and can trigger an immune response via that mechanism as well, right? So it's kind of like a triple whammy. You're going to get the cytokine release, you're going to get the flare expression being targeted by the CAR T cells, and also flare expression being targeted by like dendritic cells, making antibodies and making endogenous T cell, responses against the MHC flare. There's that triple whammy.
Right.
That might be it. But I think that oncolytic viruses also provide a triple whammy. They do cytokines, they do direct cell death, and they do viral peptide mediated immune responses. So it doesn't seem to be much different than oncolytic viruses, except for the fact that there's this massive complexity involved with manufacturing and infusing a CAR T cell, right? So it's like, it seems like you could have gotten the infection mediated cell death by just making it an oncolytic virus. But instead they said, well, what if we can kill the cells with a CAR T cell instead? It's like, why?
Well, here would be the question then, Jeff. Could you potentially have a company like Deck Bio who partners with a company like Dispatch Bio, and you just make a T cell engager for flare, and you don't have to worry about a full CAR T therapy?
Exactly. Like that would work too.
Yeah.
You would just make an antibody. That's way easier than doing a CAR T cell. It just seems like it's a solution to quote unquote make CAR T cells work in solid tumors. But that isn't like the problem. The problem is the solid tumor.
I think we are missing something. I should see if we can get Lex Johnson to come on, and he can yell at you about how you're wrong for 10-15 minutes.
Maybe. I mean, I get but the thing is like there is answers to these questions. Like for example, if you were to say, why not just make a T cell engager against flare? The answer to that question would be the endogenous T cells are immunosuppressed. So you can put in a bolus of flare targeting CAR T cells that aren't immunosuppressed, and you're going to get anti-flare immune responses that way.
Right.
So like there are answers, but the thing is like I know that there's answers to these questions, but is it actually I don't know, man. There's something that's like doesn't sit right with me for whatever reason.
Well, ideally we can get someone from Dispatch Bio on eventually to talk about this and tell us how we're wrong or how we're right. But
well, you keep using this word we. It's Ben, it's me, it's Jeff.
I'm a bit more optimistic about this approach than you are. But yeah, that being said, super interesting approach. They have been able to dose their first patient, so we'll see what happens, right? This has been the challenge with oncolytic viruses historically, is that they haven't really panned out a whole lot. And so that right there is probably the answer, right? Like, okay, Jeff, if we can do this with oncolytic viruses, then why don't oncolytic viruses work that well? And it's like, yeah, okay, checkmate, I guess, right? Like the oncolytic viruses mechanisms don't work that well, I guess, and I'd have to look into more about like what is the actual what does the field of oncolytic viruses actually look like right now? Like is it in turmoil, or is it just like doing fine? Right.
But let me scoot out for 1 second, Ben. I'll be right back.
All right. Don't go anywhere, folks. I'm all by myself. Jeff is having some issues with internet today, so hopefully we can get that resolved post haste. But yeah, in the meantime, we have been talking about Dispatch Bio, really interesting company. Jeff is somewhat unconvinced by their approach, but I had a chance to chat with one of their executives at ASGCT this past year, and I left the conversation a lot more optimistic about it, and I'm probably forgetting a couple different key details. It was very late at night when we had the conversation, and once again, shout out to Jay Rosanelli from over at Synex for setting that up. But yeah, in the meantime, I guess I can't quite change my screen here because Jeff is in charge of that. But one of the other things that we do want to talk about today is a company called AC Immune. They very recently, let's see if we can just bring them up next here.
Okay, I'm back. Sorry.
You're back? Perfect.
Yes, sir.
Right. Perfect.
What do we got?
AC Immune, Jeff, can you take us over to them?
Yes,
on the whole topic of interesting approaches taken to stimulate the immune system, AC Immune falls into the category of an active immunotherapy, Jeff. Does that sound familiar?
It does with Base Immune.
Yes, we had them on a couple weeks ago.
Mm-hmm.
And they're very interesting. Like Base Immune, they're focusing on idiopathic pulmonary fibrosis. Super interesting approach. But for those of you who are not familiar with the whole idea or concept of an active immunotherapy, it's basically almost a next generation vaccine. It's vaccines but better, where the idea being essentially instead of building some sort of specific antibody that is going to be responsible for helping obliterate some sort of problematic protein or some sort of problematic cell, can we take a step back and instead of trying to figure out what the perfect antibody is to go after some disease, instead could we work hand in hand with the immune system and essentially prompt engineer it with the instructions about what it needs to go after and get rid of, and then let it come up with its own solution. And so it's very much the idea of instead of developing your own car from scratch, I'm just going to hand it off to the engineers at Ferrari who are experts at this and get them to design the car with the exact specs that I've requested. And so what we're seeing here with AC Immune is they are, I believe it's going after Alzheimer's or Parkinson's.
So is this what you just described though? I mean, I see that it says active immunotherapy and vaccines.
Yeah.
But okay, they have a small molecule arm as well. Okay, keep going.
Yeah. But they are going after amyloids. Is it for? Yeah, it's for
Alzheimer's for sure. Yeah.
It's for Parkinson's.
Oh.
Or no, is it? Oh, okay. I thought I saw Alzheimer's. I thought I saw Parkinson's further down. But maybe I'm mistaken. Okay, so yeah, it's for Alzheimer's. So they're going after these amyloid plaques, where essentially the idea is instead of us trying to figure out what the perfect tool is for this job, we're just going to explain the task to the immune system and let it design the perfect tool for the task. And it's going to do a far more comprehensive job, and you're not going to have to worry about it getting rejected by the immune system because the immune system is the one making it. So it's a really interesting approach, and they're going after Alzheimer's with it, where we can target these problematic proteins, and the whole goal being, let's allow the experts to do the job because they will do the best job, as opposed to us trying to make a somewhat substandard instrument that's going to work for everyone. Let's just do the highly specialized work of providing the specs and allowing each person's individual immune system to create the best response.
Yes. There is one added dimension of complexity with this approach, though.
Sure.
Wild inconsistencies between patients based on their own immune system's ability to actually respond to the prompt.
Sure, but would this actually be a little bit different depending on the fact, right, that this is not a standard like viral infection, right, or some sort of bacterial infection, the way you'd think of a standard vaccine that's going after an infectious disease. This is when the immune system is a little bit more at like baseline, and there's not like this imminent attack of some sort of horrific infectious disease. It's you don't need to mount the same amount of an immune response when it's an invasion versus when it's kind of like a benign enemy within.
I wouldn't call a lot of these neurodegenerative diseases are actually like inflammatory, right? So like they are already a disease of the immune system, kind of. And I'm not super familiar with like how idiopathic fibrosis works, but I think it has to do with that might be different, but like, yes, I hear you. I think really it's like a diversity of response, right? So if we were to say, okay, we're going to make an anti-CD19 antibody, right? And we're going to give it, and that's one epitope, right? So it's going to target one single epitope of one single protein, right? Giving a vaccine is different because your body decides which epitope targeting antibody response actually is the one that's working. So it's why a company like Base Immune has a really interesting approach, because they know that idiopathic fibrosis is diverse. The pathways are diverse, and the proteins that you target are diverse. So you need to have a very like broad array of therapeutics. And how do you do that other than creating a cocktail of 25 antibodies, right? You don't. You give it a vaccine, and your body can decide what to do with it. So yeah, I like it. I just think that there's all the same nuances of like giving a vaccine. Even when you give a vaccine, people will respond differently because of the difference in their immune system's ability to actually respond to the vaccine. So whereas if you just gave an antibody, well, the antibody is going to be the antibody regardless.
Right. That being said, right, the signal here is that they've got their IND cleared and they've been given fast track designation. So especially kind of given
it is for Parkinson's. This signal here is for Parkinson's.
Oh, okay, perfect. All right. But yeah, this is the other thing too, right? I think we can all agree that the current FDA administration has been a bit more skeptical of vaccines, potentially more than is warranted. Who knows? And yet they have still seen this approach and been like, "Shoot, this looks really cool. This is important. Let's give it that sweet fast track designation."
Yeah, I would like to look into these more. Like it's all anti-amyloid beta active immunotherapy. I'm wondering, like, yeah, if you were to give like an anti-amyloid like antibody or whatever, it's targeting a single epitope and it might not like work that well. I don't know. And if you give a vaccine, like your body can create its own endogenous antibody responses, which will have an array of epitope targeting antibodies. So yeah, I mean, it's cool. That's my take.
I like it. Nice. All right, Jeff, I believe the next one is yours. Elicio.
Oh, more vaccines, dude. This is like the vaccine show here. So Elicio has been on my radar for a long time. I think before they even went public. It's a public company. They're based out of MIT. Daryl Irvine was the guy who, I think it's Daryl Irvine. Yeah, Daryl Irvine. It's research out of his lab. And we actually talked about this technology back when I was doing the innovations in waiting, Ben. Remember that, like way back in the day? What they do is they have this amphiphile platform for vaccines. Okay. Now, when you give a vaccine for and this is why I love it so much. They're cancer vaccines.
Right.
Okay. And there's a huge difference between a cancer vaccine and a prophylactic vaccine. Obviously, but there's a major difference in like the disease state between a prophylactic vaccine and a cancer vaccine. Ben, who do you give prophylactic vaccines to most often? Who gets the most vaccines?
Children.
Little babies.
Yeah.
Right. So my son is 2, and he was vaccinated against measles, mumps, and rubella.
Right.
He did not have any of those diseases at the time. Right. You give them preemptively, and then he can mount a response against that. And then if he ever gets like the measles virus or whatever it's called, he'll already have memory T cells, plasma cells, and that thing won't even stand a chance. Right. With cancer, you don't give little babies cancer vaccines unless you call like Gardasil a cancer vaccine. Right. You give cancer patients cancer vaccines, which means that the body has to take that vaccine and elicit a response immediately. Right. And go and do the thing and actively kill the cancer that's currently growing. Right. And one of the biggest problems is just like lack of a robust enough immune response from the initial vaccine like insult. Right. So what Elicio is doing, and normally, you know, what you do, how a vaccine actually works is you'll do like a subcutaneous, like an intramuscular injection of some kind of like particle, right? And local immune cells like APCs can pick it up, go to the lymph node, and present it to T cells there and do the whole thing. But that process is not super robust.
Right. And kind
better, a better way would be to just get those particles into the lymph nodes. Like take the actual cancer vaccine and just put it into the lymph nodes, right? And what Alessio does is they take a peptide-based cancer vaccine and they'll strap on albumin onto the cancer vaccine. And what albumin does and what albumin is it's big, and it will promote lymphatic trafficking of the vaccine, and it will just shoot right to the lymph nodes, and then you get high concentration of the vaccine inside of the lymph nodes, and you can expedite and make that process way more robust. So they'll take, they'll take like a mutant KRAS peptide, strap albumin to it. Oh gosh, they'll strap albumin to it and infuse that, and then it goes to the lymph nodes, and it will activate local cells there, and it's just better. It just works better as a vaccine. So that's what they're doing, and I think that what was the most recent signal? Oh, phase 1 investigator-initiated trial opened at Memorial Sloan Kettering. Yeah, so I think it's in I'm pretty sure it's in pancreatic cancer. KRAS, it's a KRAS pancreatic cancer vaccine. Yeah. So, yeah, thoughts?
Interesting. So, back up and help me understand for just a second. We're putting this into the lymph nodes. Is this basically just like a faster response play compared to?
I think it's faster. Vaccine? It's faster, but it's ultimately more robust.
Okay.
I think you can have you'll have more APCs pick it up. You'll have a broader or a more robust T cell response and faster. So I think that it's what cancer vaccines need. You need a basically immediate and a very robust immune response, right?
So it's almost like, if I was handing out 100 pamphlets, it's like, well, if I go into the middle of a residential neighborhood where there's one bus stop once per hour, it's going to take me a long time to hand out all of the pamphlets. But if I go to a central terminal of the city, I can hand out all those pamphlets in 5 minutes.
Right. And with the bus stop analogy, you would eventually get those 100 pamphlets handed out.
Yeah. It
right.
Just takes so much.
But it'd be scattered. It would take a long time. If you went to Central Station and just threw them around, you'd get way more people and faster.
Yeah.
Or I guess you'd get it faster and bam, a big crowd of people. So yeah, that's the idea.
Super cool. Okay.
Mm-hmm. And what the thing that I mentioned with the therapeutics in waiting was instead of strapping albumin to a cancer vaccine, they were strapping active sting to a cancer vaccine. So the active sting was big enough to mimic the effects of albumin and help traffic to the lymph nodes, but then it wasn't an inert molecule like albumin, it was active sting. So it would be taken up by APCs, and it would activate the sting pathway, creating, inflammatory responses in the lymph node, and you could get an even more robust immune response. I'm pretty sure that innovation is still sitting in the MIT tech transfer portal.
Okay. Well, that would mean then maybe this is now a popular and trendy therapeutic application to go after. And if you were to take that tech transfer out of MIT, you would definitely want to have a bunch of investors. So let me tell you about Tetri Bio. It's all about who you know, and now you can catch prospects and investors the moment they're ready. Deep company research, live signals from your socials, verified contacts, and a whole lot more. Built for biotech and no one else. That's Tetri.bio. What do you think, Jeff?
TETRI.bio. Yes. Tetri.
It's a company that
go check it out.
Excellent. All right. So check that out. All right. So Alessio, super cool. Interesting approach. So if we move on from there, Jeff, I believe the next one here is Silence Therapeutics. I believe that one's mine.
I have no clue what this is, so enlighten me.
Silence Therapeutics. So this one is pretty interesting. I also am going to have a little bit more. I'm going to consult my notes here a little bit because this one's really interesting, and my silly monkey brain was a little bit confused. But
it's
basically
called Unga Bunga brain.
Unga Bunga brain. Yes, that would be the more accurate version. Okay, so basically we're dealing here with a blood cancer where the bone marrow becomes obsessed with making red blood cells. So you just have an overabundance of red blood cells. Who would have thought that you could have too much of a good thing? And basically it's this particular activating mutation in JAK2 that keeps growth signaling switched on, so red blood cell production keeps running when it should slow down. And so basically this is going to be a play to basically turn that off. And so they have an siRNA that they've developed, and they've kind of got a whole program around it that's designed to very specifically go in and like tamp down overproduction of red blood cells. And so like I didn't even know that this kind of disease existed, but apparently it's
polycythemia vera.
Dude, there's just so many things that can go wrong. Like, we are not thankful enough.
Don't even get me started down this rabbit hole. You have no idea. Studying molecular biology is ridiculous.
It is equal parts, amazing and terrifying.
Mm-hmm.
To just be like, yeah, we are fearfully and wonderfully made. No,
you're like one atom away from dying of cancer all the time.
Yeah.
So, but don't worry.
Well, what's the I was listening to a podcast the other day, and some guy was like doing an ad read, and he was saying, so I looked up the stats in your area because he was selling life insurance or something.
Mm-hmm.
Like, I looked up the stats in your area, and I'm sorry to tell you, no one made it. Everyone dies eventually.
Oh, I was like, I don't get it.
But anyway, so what this company is focusing on, right, is RNA. They're using something called GalNAc. It's useful because it directs these siRNA very efficiently into hepatocytes in the liver. What's inside those cells. The siRNA uses the natural RNA interference machinery to just destroy that messenger RNA that's going to stimulate the overproduction of the red blood cells.
Do you know what the difference is between an siRNA and an antisense oligonucleotide? An ASO? There must be some kind of difference in like the machinery that they recruit. I'm gonna guess I know that like the siRNAs recruit like the dang it, like Dicer and whatever, I forget.
I feel like because siRNA like forms a loop. I don't believe that antisense oligonucleotides do. So that's probably the major component there. Is basically you've just got this tiny little section on the end that's matching a sequence versus I believe an antisense oligonucleotide is very much more like a protein complex that's attached to a single strand that doesn't have a loop in it. I could be wrong.
That doesn't sound right. But it's got to be with like the actual like molecular mechanism. I don't know. But they both do the same thing. They both target mRNA for degradation, siRNAs and ASOs.
Between an siRNA and a oligonucleotide antisense. Okay, small interfering iRNAs are double-stranded RNA molecules that trigger gene silencing. An antisense oligonucleotide is a single-stranded DNA or RNA chain. I was kind of right, Jeff. I said it was a loop, and therefore it causes it to become a double strand for siRNA versus a single-stranded for the antisense oligonucleotide. Eat your heart out, Jeff.
I don't think that's what I said was wrong. You said it's a protein complex that's going to bind a small piece of tail RNA that sticks out. I don't know about that. Anyways, that's fine. I was kind
of right. I was also kind of wrong.
Silence. I feel like I looked into Silence. They have a really cool way of like delivering these things. It's very interesting getting the technology behind these like RNA-based therapeutics. A lot of the actual like core innovations have to do with how they get these things into the cells in the first place. A lot of them will like strap them to antibodies or strap them to, you said that GalNAc, right?
Right.
There's a lot of like really cool companies doing it really interesting like RNA fusion technology to like strap an siRNA to an antibody that then gets like cleaved once it's inside the cell, and it's super cool. So yeah, I never understood why there's not any, or there's not as many like siRNA based therapeutics for cancer. because one of the biggest problems in like cancer therapeutics is this class of so-called undruggable targets. Right? Like transcription factors or whatever. It's like, okay, then just use siRNA to downregulate the mRNA and just like stop the expression of this thing. Like, why can't you just do that? Right? But
I think it's probably got something to do with the fact that cancer is very well known for mutating.
Yeah, but I think that it would not really in that respect. Like, it has to do with getting the thing to the cells, I think. I don't know. What do I know? I don't know a lot of things.
You heard it here, folks. All right.
Jeff doesn't know anything. Let's do a thought experiment. Ready? You've got KRAS, mutant KRAS, G12V KRAS mediated like pancreatic cancer, right? Just administer the mutant KRAS specific siRNA systemically. If the only cells that have the actual mutant KRAS are the cancer cells, then the mutant KRAS targeting siRNA won't touch anything else.
I feel like that's
so just
really
get it into every single cell.
Because if you mutate that just a little bit, then you're free.
No, that's not how it works, though. I mean, there's that specific mutation is what's causing the protein to become oncogenic in the first place. So if it mutated, like that isn't like a problem. Like there's EGFR targeting small molecules that, like, yes, you'll get like first generation EGFR mutations, second generation EGFR mutations, but I'm not super sure. Like, I don't know.
Are you able to precisely deliver that to just the cancer cells well enough?
You wouldn't need to, because it would only target the mutated KRAS, like mRNA.
Yeah.
So if you did it systemically, there's got to be some more nuance to it. Maybe it's like dosing. Like, how do you get that siRNA systemically? Right. You would need to have so much of it. It wouldn't be feasible. I don't know.
Gotcha. Okay, interesting. Well, that being said, Jeff, $100 million is our next story. $100 million in a series flipping B. We should.
Arika?
Yes, Arika. $100 million with an M.
Money.
$100 million. And what is the indication they're going after here?
So I don't know. This was super cool because they had a purple logo. Not gonna lie to you, it's stuck out in my book. They raised $100 million. AI-based drug discovery, lab in the loop engine, integrating high throughput single cell functional screening and generative AI to redesign drug discovery end to end.
Oh, okay.
You know what, Ben? It's about time that I looked more into like how these AI models actually work. Taking single cell data and then using quote unquote using AI to design drugs. Like, I get the concept. I can kind of see like through the smoke how that would work, but I have no idea how it actually works. You know what I'm saying? Like, how do you use the AI to? And I think I asked Kevin Walton this. He's the CEO of Base Immune, because he had posted on LinkedIn about how they use AI for their like drug discovery process.
Right.
And I was like, where do you use AI in the process? Is it at step one, where you take a bunch of like, I don't know, pathological molecular pathways, and you can use AI to like figure out which of these targets are driving the pathology that are actually targetable? That's like step one. Step two would be like, I guess, designing the therapeutic itself and like optimization of the formulation. Right?
Yeah.
Where do you know more about this stuff? Like, where does the AI actually come in?
Because I think primarily it has kind of been on target discovery, and then for a company like Base Immune, right? Like, they can kind of stop there. Because the
and I think that
is to kind of like identify the epitopes and antigens that you want to go after that are going to be most effective at.
Oh, and they just use an active immunotherapy.
Yeah. I believe they're primarily using AI just to kind of identify what are going to be the best targets that we kind of point the immune system towards.
Yeah, and like using single cell, like high throughput single cell functional screening, that just like screams like molecular pathway analysis. Like, what is the pathology being driven by? And we can just get a bunch of like single cell RNA sequencing or like single cell mass spec to figure out like, here's this pathological cell or whatever, and we've got this trend of like really powerful signaling in like the, I don't know, AKT pathway. and like you can use AI to dissect those things. That makes the most sense to me. But I'm ashamed to say I left lab right as AI was like really coming online. Right. And I'm more of like an AI and like business development guy. I'm not an AI and like biotech, like biology. So actually let's do a little bit of exploring. Aurica. Let's go to their technology page and see if they have any like diagrams that will allude to this. Okay, single cell, fine. Okay, here you go. Functional screening, big data. So the functional screening produces big data, of course. That big data goes into an AI model. Autonomous evolution. I'm sorry. That's a curveball. Okay. High throughput synthetic biology technologies to mine, synthesize, and characterize therapeutic candidates at scale. Okay, there it is. There it is. Characterize therapeutic candidates. So they're finding the targets.
Right.
But the autonomous evolution, that seems a little bit creepy to me. What is that? Like, what even would that be?
Those are two words you don't usually think of putting together.
Yeah, autonomous. I guess, isn't all evolution autonomous? I guess, kind of. Drug expressing cell, reporter, that's fine. Millions of reactions, single cell. Oh. Okay. We employed geometric aware protein language model and reinforcement behavior or learning to drive the generate, test, learn, and optimize cycle. Oh, it's okay. With unprecedented data iteration efficiency, our AI solutions allow accurate and rational therapeutic design with desirable attributes. Yeah, that's super cool. Yeah, you know what, dude? We got to have like some kind of incentive to spend some time learning about how these platforms actually work, because we're going to see so many companies have these. Like everyone's going to be able to build these things internally.
Right.
It's the future of medicine, man. It really is.
And they're based in China. I was going to say it'd be interesting to see if we could get one of their executives on.
Someone's
in California.
A lot of the Asian countries will have like a U.S. base, like a U.S. office as well.
Well, that means they might have someone over in California then who'd be interested in
coming. Yeah. Which how like, okay, they're post Series B now, I guess, right?
Yeah.
Oh, it was today.
Yeah, it was recent.
Dang.
Congratulations to them.
The press release talks about what they're going to do with this. Let's see. Will direct proceeds primarily toward research and large-scale training of its next-gen biological foundation of models. Oh my gosh. You need $100 million for that? I guess they're probably doing a lot of like proprietary data set generation.
Yeah. Those data centers aren't going to grow themselves.
Yeah, seriously. Which that'll be huge. I feel like this is what this, it sounds bad saying this, but I feel like academia will be really good for that kind of stuff. Like just like creating a bunch of data, but then you're pretty much just like relegating academia to being like lab monkeys.
Data junkies.
Go in here and just like crunch the numbers. Like just make us a bunch of data that we can then go and just like buy.
Yeah.
Yeah. Okay, that makes sense.
Cool.
And what are they doing it for? Normally just a lot of stuff. Cardiometabolic, inflammatory, and CNS indications.
Pretty standard.
GPCRs. Yeah, dude, GPCRs suck. They're like so hard to target, and they're like involved in so much pathology. In fact, there's a whole company called Antiverse that's doing.
We talked about them before, right?
We did a long time ago. Yeah. They're doing AI-driven platforms to design antibodies against ion channels and GPCRs. Because because
it's the OG.
It's like really old. Yeah, like they got $9 million Series A. This is what's interesting though, right? Antiverse has a $9 million Series A, and they're doing kind of similar stuff, like AI based. Although they're doing like antibody, not so much. Okay, this is the difference. Companies like Antiverse are doing AI based, actual like therapeutic optimization.
Yeah.
Right. Whereas Aurica is doing candidate discovery.
Right.
Completely different uses of AI.
Yeah.
Yeah. Because we already know the GPCRs that we want to target. We now need to like optimize antibody formulation to target them.
Cool.
Yeah. Anyways, what is next? Who we got?
All right, the final one of the day before we round out the show is kind of out of left field, Jeff. This
Okay.
one is a company called
Oh,
Magnendo.
yeah.
Magnendo. This is
Robotic
kind of
navigation.
I'm actually really interested to explore this a little bit more, because this is now talking about like how are robotics and AI going to converge into healthcare. And so this
Okay.
company landed $32 million in the form of an ARPA-H grant to go after robotics that are going to help address stroke. And so the whole goal is someone has stroke, all of the interventions that take place after. With doctors, it's a little bit hit and miss, right? It's difficult to say somewhat consistent, but what if you do this entire thing with robots?
Okay.
And so they got $32 million to be like, let's freaking take some robots and throw them at stroke.
Yeah. With and they're, I'm going to guess it uses AI as like part of the guiding
I believe
mechanism.
so. But yeah, it's spun out of MIT. Like this is no
Yeah.
joke. It's not just some guy, in Kansas being like, hey, I got a robot in my garage. Like, this is out of MIT. So this
I'm sorry, Ben. Are there a lot of guys who are in Kansas with robots in their garage? What do you think of the United States? Y'all want some
beautiful
robots?
people who have tigers in their backyard.
Okay.
And
That'd be like me being like, yeah, all them Canadians have that maple syrup in their garage.
I've got it in my fridge.
Yeah, exactly. I have my 14 fridges full of maple syrup.
I have one fridge. In fact, my wife is further down. She grew up closer to America, further down in Ontario than I did, and her family grew up with like Aunt Jemima's like table syrup.
Okay.
And I was very offended when I first started coming around her family, because there is only one gold standard for syrup that's put on pancakes, and that is maple syrup.
Yeah. If it ain't from a tree, it ain't for me.
Amen. 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 None of this high fructose corn syrup crap. How did we get here?
I have no idea, actually.
But anyway, no, I just thought it was interesting, given that I really like ARPA-H. If you guys aren't familiar with ARPA-H, like, literally take 5 minutes to go Google them and see what they're doing. We've been able to interview a couple different people inside of the space, inside of the ARPA-H kind of cohort of companies. I love to see what they're doing. It's basically like pragmatic moonshots for really interesting, swing for the fences biotech. And so they're funding everything from AAV getting made to
AAV
plants,
and tobacco.
yeah, AAV and tobacco plants to stuff like robots for stroke recovery. So it's really interesting to see stuff that is too risky for like a VC to necessarily take on, and yet you can kind of like tap into the deep pockets of government to be like, hey, we will give you the money. You've got to meet very specific guidelines and deliverables, but we will give you the money to go see if this thing works. And if it doesn't, no harm, no foul. But yeah, it's pretty cool. Excellent. Well, Jeff, I think we could leave it there. I don't have anything else that I wanted to talk about.
I can't believe that I've been frozen for like an hour. Can you believe that? This is insane. It's so embarrassing.
You need a new computer, man. Or
it's my internet. It's the I'm on my phone's hotspot.
That'll do it.
I'm traveling.
You're traveling. You gotta get some Starlink. They have those backpack Starlinks now. You can just set up wherever.
That's nuts.
Yeah, you should get one. Excellent. But ladies and gentlemen, thank you so much for joining the Biotech Voyager today.
My stupid face.
What's the matter?
My face is just like boo.
Thank you so much for joining the Biotech Voyager today. We had a really great conversation at the very beginning of the episode, if you're just joining now, with a gentleman called Thane from Deep Science Ventures over in the UK. Really fantastic conversation about how they are developing a PhD program or a doctorate program that ends with a company getting generated. So instead of just kind of becoming a single operator inside of a niche area of science, the end result of your doctorate degree is a company inside of a breakthrough component of science, technology. Super cool to look at. So definitely go check that one out. All right, folks, we're going to leave it there. Thank you so much for coming. If you have not subscribed, please do go ahead and subscribe. We would appreciate that immensely. And until next time, we'll see you later.